Thank you. Thank you for giving the opportunity to present my work. Actually, I'm a second year PhD student, and currently I'm working on to develop aptamers for a small molecule. And my title of the topic is the high throughput sequencing using HT-SELEX and by combining the computational approach to generate a specific and sensitive aptamer against the carbendazim. This is my overall outline of the work. So first I introduce what is the carbendazim, why I chose the carbendazim. As the carbendazim is comes under the category of the pesticides, it's a widely used fungicides in India all over the world, but especially in the developing countries had high uses of the carbendazim, and the developed countries already banned these carbendazim molecule. So there is a huge burden on the trade rejections because by looking the agriculture trades, because the presence of these molecules, there is a high rejection of economical burden. So why this carbendazim molecules causing the problem? Why is it causing the problem? There is an n numbers of the things that is associated potentially with the carbendazim molecule. First, it is causing the phytotoxicity. It's causing the phytotoxicity, and it is comes under the human carcinoma. And the WHO has already categorized these molecules. It's not in a certain MRL maximum residue limit is allowed in the food and grains. So the food and agriculture organizations only looking these molecules should be used in a permission limit in the food industry. So the other potential hazard which is causing by these molecules, such as the in our food commodities, fruits, vegetables, soil, and water. So it is a great impact on the environment. So now it's come into the picture. So how we can use the aptamer to diagnose or detect these molecules, because it is because since it is a small molecule, it's require various high-end instruments just like a HPLC and the other high-end instrument, which is not feasible. So we are using the aptamer and develop a low-cost rapid diagnostic kits. So as you all know, that the aptamer is a single density molecule, and it's when it binds to the target, it's fold in a tertiary structure and make a complex. So now the comes in the picture. So I have performed the SELEX. So in the SELEX, I have used the gold nanoparticle. So let me know why I use the gold nanoparticle. This is the overall pictorial representation of the my SELEX technology. So the first, as you can see, the pesticides molecule and the aptamer come closer to in the contact, and we adding the gold nanoparticle. Because why use the gold nanoparticle is also known as the gold SELEX, because in it is a non-immobilized method. There is a no need of to modify the aptamer, either aptamer or the small molecule. So there is a number of epitopes available for the interaction to the aptamer is high. So, and we do the centrifugation and elude the non-binders aptamer. And after that, we perform a asymmetric, because for the next cycle, we need a single stranded DNA molecule for to generate to go jump into the next cycle. And this process goes until the enrichment of the highly specific aptamer. So for the enrichment, we follow the simple polar particle assay, apart from the other assays such as the Go fluorescence assay and and the RT-PCR based. Some of the RT-PCR method we will be talking in the later part. And we also adopt the NGS and the characterization of the aptamer. Apart from that, the characterization of the matter, we also some using the computational technique, because there is a lot of aptamers already available. Their sensitivity and KD is very low. So how we can use the computational knowledge that can be built in that sense, that can be increase their affinity and specificity, especially in terms of KD. So my end goal is to develop a simple kit. Like right now, I am here. So I'm determining the specificity and the sensitivity of the aptamer. That is, I'm just generating through the SELEX process. So for this, we have synthesized the gold nanoparticle. Just as a state reduction method is already established method, and we also synthesize the graphene oxide and their characterization. So we got the uniform gold nanoparticle and the monolayer sheet of the graphene oxide. Their FTR and functional properties we have categorized. Okay, now this comes in the picture. So the main The main hook is the enrichment. How to track the enrichment of the SELEX? Okay, whether you are going in right way or in a wrong way, and where we to stop ourselves to do. So for this, so enrichment of the SELEX, actually we perform a Go assay, which is a fluorescence assay. So we have performed a asymmetric PCR method. As you can see that with a, there are different cycles. This is starting from one cycle to the 10 cycle, because I have end up with the 10 cycles. So this is a green one which is show the single strand DNA, because we have tagged these the primer with the fluorophore molecules, and the above one is the double stranded DNA. We do not require the double stranded DNA. We just remove it and we elute the single strand DNA for the fluorescence assay. So we have used the Go assay with the graphene oxide. So in the, as you can see here, the zero cycle serve as a control, and as you know that the enrichment is happen in the SELEX is onward of the fourth cycle. Usually there is a high chances, because of the initial cycle there is a random aptamers molecules, so the enrichment start from the fourth cycle, and we track with the enrichment with the fluorescence assay, and we also perform a counter SELEX at the 8 cycles. So before the counter SELEX, what we have done, we using the carbendazim, the similar molecules of the carbendazim, and some of the cross-reactive binders, so that there, because we are trying to develop a kit which can be used in a food and feed commodities, there are some cross-reactive molecules. There are also chances that these aptamers bind to these cross-reactive, because I heard about that there is so much that the cross-reactivity is the major problem with the aptamer, because so we perform the counter SELEX at 8 cycle. As you can see that 8th cycle, there is a sudden decrease in the fluorescence, which is represented in green, and we continue 2 more cycle, that's the enrichment. So there is a fold increase in the enrichments from onward from the 4th cycle, and apart from that, we also perform the gold nanoparticle as a standard method. So as you can see that in the 0 cycle, there is a no disaggregation. As we perform the gold nanoparticle, the property of the gold nanoparticle is that it is bind to the aptamer. When the target is present in the solution, the aptamers leave from the gold nanoparticle and it bind to the target. So the gold is free, and we use the salt that's aggregate the gold nanoparticle. So by using a simple method, we can track the enrichment. Now, apart from that, no, it is not only that you have to just using a standard method. There is a RT-PCR based method, which is very known method, which is highly used. So as you know that the initial library, initial SELEX library, which is contained the heteroduplexes. So the heteroduplexes, as the SELEX is processed, as the enrichment is happening, these heteroduplexes come in the homoduplex. So there the melting temperature, their melting temperature is the stringent. So we track these homoduplexes with the help of the RT-PCR method, and there is the amplification plot. Apart from that, as you can see that these the library, the CT value is high, as the library which is usually don't have a aptamer for the carbendazim molecule, so there the CT value is high, because the CT values indicate that there are high template for the small molecule. As the cycle is progressing, there's a decrease in the CT value. As you can see that up to the 6th cycle, there is a decrease in the CT value. When we introduce the counter SELEX, there is a jump in the CT value, which shows that some of the R aptamer is bind to the some cross-reactive molecule, whether we introduce in the counter SELEX. So then we perform, we also perform after that counter SELEX, we perform 2 more cycles, and there is a now gradual decrease in the CT value, which shows that there is a still aptamer that is bind are the small molecule. Now, after performing the enrichment, that we show that that's we getting the enrichment, we getting the clue that yeah, we have a aptamers where we got the enrichment. Now this is coming in the picture of the HT-SELEX. So for the HT-SELEX, just we did the symmetric PCR, because for the we did the symmetric PCR, we using the adapter to ligate, so that the machine can read the sequences. And this is the overall the HT-SELEX procedure that we adopted. And for right now, we have do the HT-SELEX of the 0 cycle as a control, and the HT-SELEX of the 10 cycle, because we there's a high aggregation in the 10 cycles. So we did only the what is the raw reads? So there are 34 lakhs raw reads we got, and in 10 cycle there are 40 lakhs reads, and total basis is there in billions. and what data we use is around a 1 GB data. Okay, now we using the aptamers. The aptamers is a very good software for the enrichment or the tracking of the HT-SELEX data. We using the aptamers. So as you can see that in the zero cycle as a control cycle, uh, there is no any enrichment is happening. So there is only a single tone, and we can also see there is a frequency. There is only 25 and 21 and very limited number of counts of the aptamer, because the zero cycle do not have a high number of the binders for the small molecule, and these are the frequency of the ATGC. And because our random library, because we targeted the random library with the N40, so we got the highest peak of the N40. Now it comes in the picture that in the 10 cycle, as you can see that in the 10 cycle, there the counts of the sequence count is the increase exponentially, and we got a count in thousands, and we are in this process. We are finalizing the aptamer, because we are already in the process of synthesizing the some of the aptamer for their binding affinity and the specificity and KD determination, and we got a very good frequency of the occurrence around 22 lakhs, and these are the distributions of the ATGC values. Now, the actually right now we are in under process of working. Actually, we are now determining the KD, and after sequence that we don't know. So we right now we have not shown the data here, because right now we are finalizing the which of the best aptamer, because we are trying to patent this aptamer, so that we are for this reason I'm not showing the sequence, but at the KD is not finalized at all. So that's why I'm not showing the further data. Apart from that, the in vitro SELEX method, as you know that it's taking a too much time. So apart from that, as the sir told, the various speakers also told about the computational method. So we also explore the computational method by using the already standard methods. Already that is a standard aptamer is already available. Why we not choose to increase the specificity and sensitivity? In that way, we cannot do the SELEX, because already the aptamer is available for a particular types of the target. Why we are repeatedly doing the SELEX? So we generate a pipeline. This is the pipeline in which uh, we explore how can we modify and how we can generate the aptamer and improve the sensitivity and specificity of the aptamer. For this reason, we use the standard aptamer. We use the 6 standard aptamer, which is already available for the carbendazim molecule, and their names is CZ1, CZ2, CZ5, and this carbendazim molecule. And this is the secondary structure we draw with the help of ampoule server, and this is the overall pipeline. As you can see that the so we first we got the sequences, then we added the primers. So we just to reset the roles of the primer, because the primer binding site is not in the part of the picture when the small molecule interact with the aptamers, and we using the ampoule to get the 2D structure. Then we using the some RNA composer tool to get a 3D structure, because for the MD simulation and some kind of something we need a 3D structure of the aptamer. So there is no standalone method, or there is a crystal structure is not available for to determine, because there is no PDB structure of the aptamer. So we generated this pipeline to create a 3D structure of the aptamer, and once by using some manual modifications and some of the Python code, we develop a 3D structure of the aptamer. So this 3D structure of the aptamer and the hook is that we perform ensemble docking. Is the earlier docking is when we just get a 3D structure of the aptamer and you start the docking. But in the ensemble docking, what we perform a MD simulation for 100 nanoseconds, and we got a 10 most popular ensemble of these clusters. When these clusters, we got these clusters, we adding a multiple small molecule, because we don't know where the small molecule bind to the aptamer. So as you can see that here is a number of small molecule. There is a predicted binding site. Some of the molecules bind from here, here, and here. So we don't know which is the binding site. So we perform an ensemble docking, and after getting these batch scores, we just doing the MDS. So during the MDS, what is happened? This is a low affinity sequences. Suppose that this is not a target binding site for the small molecule. These molecules leave from there. Only the affinity regions where the small molecules bind with retain, which is studied with the help of the MD simulation. So this is we got the highly stable, and we finalize this is the region where the small molecule, where the carbendazim bind. So what we got from this information, we got the information that this is the we modify this region, we can increase the affinity and the specificity of the aptamer. Maybe it will be decreased, but we have apart from that, it will also help in the truncation, which will reduce the cost of the aptamer. So for the MD simulation, this is the picture of the system preparation and the condition which I used. I used the software GrowMax and all under char 36 force field cubic box and the salt concentration and the temperatures. And as how you can process that the simulation is going the valid. So we have tracked the trajectories analysis. So for the trajectory analysis, we conclude the RMSD because as a small molecule, as the aptamer interact with the small molecule, it become a complex and compact. So their RMSD is reduced, and it is recognized as small molecule or any kind of a target. And there is fluctuation of the root mean square fluctuation, which show the fluctuation. So as the small molecule binds with the aptamers, their fluctuation is reduces. So we track that the fluctuation reduces that the aptamer is binding to the target sites. So this information we got from the trajectory analysis and the radius of gyrations and the SASA. The radius of gyration is a measure the compactness of the aptamer, and the SASA which shows the solvent accessible surface area. As the aptamers interact with the molecules, it become a compact, and there is a reduce in the solvent accessible surface area. So this information is just giving the clue that your simulation is valid, or if it is happened something else, we can stop and we look further. Now, so and as already told that we use 6 aptamer here, that is the CZ1, CZ2, CZ5, CZ6. This is the hydrogen bond occupancy. As you all know that the aptamers majorly interact with the small molecule with the non-covalent interaction. Especially the hydrogen bonds has a crucial role in the aptamer recognition. That's the other interaction. So we track the hydrogen bonds. So as you can see that after the splitized force, after 100 nanosecond, we got this is the binding region. That's the small molecule, the carbon interact here, and we zoom in, and these are the number of nucleotides which show the high hydrogen bond occupancy. So if suppose that this is a A28, if we replace this A28 with the other nucleotide, and with the hydrogen bond occupancy is increased, we increase the sensitivity and the KD values of this aptamer, and we doing this for the other aptamers also. So there is a vary in the hydrogen bond occupancy, as you can see here. Or as one of the most important information that we got from this study, that's these single molecules only have one binding region. There is only there is bind to the one binding region, which show that only the single pocket which is involved in the binding with the aptamer. So these are the hydrogen bond